Fats are a highly efficient store of energy, but there is a snag. It is really easy to turn starch or glucose into fats, but only about 6% of the fat, by weight, can be converted back into glucose. This can cause problems during long-term starvation in providing sufficient energy for the brain. To understand this, we need to look a little more closely at their chemical structures. Fats are esters, formed by reacting long-chain fatty acids with glycerol. It is easy to convert glycerol and glucose, and easy to turn glucose into fatty acids, but it is impossible for animal cells to convert fatty acids back into carbohydrates, no matter how hard they try. Plants can do this trick, and it is very important in germinating seeds, but animal cells have either forgotten how to do it or they never mastered it in the first place 2,000 million years ago. This means that all animals must be really careful to conserve their carbohydrate stores. Blood glucose is an excellent source of short-term energy, quickly mobilized and easily used. It is able to support the highest levels of athletic performance, and it is essential for rational thought. But it is too bulky for long-term storage, and it very quickly runs out. All this turns blood glucose regulation into a rather delicate balancing act. If we eat a high carbohydrate diet, then we cannot store large amounts of glucose because it exerts too much osmotic pressure. Neither could we lay down sufficient glycogen stores because that takes up too much room. Instead, we have little option but to convert most of our carbohydrate into fat. Fat cells, or adipocytes, can do this by using the Krebs cycle as a metabolic clearinghouse. The twin processes of glycolysis, which means the breakdown of glucose, and lipogenesis, meaning the synthesis of fats, are both promoted by the hormone insulin, which is mainly secreted after we have eaten. Much as we might like to, we cannot stop this happening. It is hardwired into our metabolism. The only way to prevent this rapid conversion of dietary carbohydrates into stored fat is to starve ourselves and eat less food. Between meals, opposite processes come into play. The hormones glucagon and adrenaline promote the breakdown and mobilization of fats, and the free fatty acids are released from the adipocytes into the bloodstream. They provide a ready source of energy for our heart, our kidneys, and most kind of muscle fibre. But unfortunately, it is very difficult to get them into the brain. This is because free fatty acids travel in the blood stuck onto a protein molecule called serum albumin, and there is no system to transport the across the blood-brain barrier to power conscious thought. Fortunately, adipocytes also release the 6% glycerol left over from splitting fats into the blood. Glycerol is taken up by the liver and converted into blood glucose through the process of gluconeogenesis. This means the synthesis of new glucose from a wide variety of precursors, including glycerol from adipocytes, lactate from exercising muscle, and most of the amino acids found in proteins. We shall return to the amino acids in a few minutes' time. This system can only work efficiently if most tissues normally prefer to metabolize fats. This is exactly what happens. Between meals, under basal conditions, the adipocytes gradually hydrolyze their stored fat and release a steady dribble of fatty acids and glycerol into the bloodstream. Muscles utilize the fats, and the liver takes up glycerol and turns it into blood glucose. Most tissues ignore this glucose because under these conditions there is very little insulin around. Insulin is widely needed for glucose entry into cells, so the available glucose is mostly reserved for the brain, which doesn't need insulin for glucose uptake. Gently exercising muscles prefer to oxidize free fatty acids, and in any case they cannot take up glucose because there is very little insulin around. If the animal becomes more active, then the autonomic nervous system increases secretion of adrenaline. It is likely that glucagon will rise as well. This in turn increases the rate of fat mobilization using a hormone-sensitive lipase that is present in fat cells or adipocytes. The same hormones simultaneously increase the breakdown of glycogen in the liver, so more glucose is released into the blood. The way this is achieved is rather beautiful, but we do not have sufficient time to deal with it today. If you took some blood samples, you could see the glucose start to rise, but most of this is unavailable to tissues because there is little insulin around. Only the vigorously exercising muscles are allowed to take up glucose without insulin, so the resources are directed towards those cells which are actually putting in the work. Glucose is more easily metabolized than fats, so vigorously exercising muscles can deliver much better performance than is possible using fats on their own. At moderate work outputs, most of the glucose is oxidized to CO2 and water in order to make ATP that will power muscle contraction. At the very highest workloads, we exceed the aerobic limit and our muscles start to recruit rarely used anaerobic fibers where glucose is broken down to lactic acid and oxygen is not involved. This is a hideously wasteful process and we can't keep it up for very long. Anaerobic muscles quickly become fatigued, but for many animals, those last few enormous, powerful contractions can make the difference between life and death. Fortunately, our liver can come to the rescue once again. The lactic acid, also sometimes called lactate, released from anaerobic muscle fibers, is recycled by the liver back into blood glucose, using the gluconeogenesis pathways mentioned earlier. These two organs, liver and muscle, cooperate with one another, and the complete circulation is called the Cori cycle, after the husband and wife team who first discovered it over 80 years ago. So far, we have mainly discussed carbohydrates and fats, but a normal balanced diet also includes proteins as a major energy source. Sometimes you will hear proteins described as bodybuilding foods, suggesting they are used for growth and muscle development. But compared with our total intake, only tiny amounts are used in this way. If you increase your protein intake, then your biceps muscles get no bigger, much as you might want them to. Most of our dietary protein is broken down to its constituent amino acids and used for energy to generate ATP. This may occur directly or through conversion of the amino acids into fat or carbohydrate as an intermediate stage. Once again, the reverse process is not possible. There is no way that our bodies can take inorganic nitrogen and use it to convert either fats or carbohydrates into proteins. The best that we can manage is a very limited interconversion of a few very common amino acids into one another, a process of robbing Peter to pay Paul. In theory, this might allow us to rebalance an otherwise unbalanced diet, but it would have to be a very peculiar diet before it was any real value. There are, however, two sets of circumstances when it is advantageous to convert proteins into carbohydrates. The first is during starvation, as our muscles gradually waste away, when almost all their constituent amino acids are recycled into blood glucose through the process of gluconeogenesis, mainly in the liver, but assisted by the kidneys and the gut. Both these secondary sources share the ability of liver cells to export glucose to the rest of the body. They require this ability in order to perform their normal physiological functions of recovering valuable resources from the urine and absorbing nutrients from the diet.